There we go. We're recording. So, like I said, Michaela is physically here. Redmond is online. We got Van Cross online. Tudor. Tudor is here. A little extra credit for being physically here. Rice is here with a little extra credit. Wait, a lot more extra credits. You guys all have WVSU gears. Okay, so for you guys is 2.6 as opposed to 2.5 as opposed to 2.4. So even more extra credit. Midkiff is here. NWVSU gear 2.6. Sipple is online. Let's see. I see Reed is online now. I also can see from my screen vibrating or looking like it's vibrating. I can tell Reed is trying to say something. Sorry, I can't hear you. I don't know what the issue is. I was just listening to Bob and Tom earlier, so I know the speakers work. Anyway, yeah, I can see you're talking. All right, look. So Reed, since I can't hear you, if you take your camera off and use sign language, American Sign Language, then we'll be okay. And I'm just kidding. I don't know American Sign Language. I only know a few things like turtle and thank you. Of course, Kinsey's here. And Massey is here. So extra credit for physically being here. Newton is online. So I think I have everybody who's currently here. Redmond, Sipple, Van Cross, Reed, Newton online. Rice, Tudor, Midkiff, Kinsey, Kayla, and Samuel. All right. Good morning. Richardson is here in person. Again, extra credit. It's not much, but it all adds up. All right, we have less than 2 minutes. Guess I'll do announcements. Now we have a minute because it's being recorded. So hopefully, if anybody shows up, after I do the announcements, hopefully they'll still be caught up. Oh, I said Richardson. I'm sorry, Hemmings, right? Because now we have Richardson. I'm sorry about that. Hemmings is here with a little extra credit. Richardson's here with a little extra credit. So announcements. Well, I definitely do this one first because I've already put it in writing, so I'll say it again. This is the last chapter, right? Chapter 7 is the last chapter of the exam. The next exam is going to cover chapters 5, 6, and 7. It's all about energy, right? All about the transformation of energy and how cells need energy, that property of life. Anyway, so when's the exam going to be? I don't know. That'll be up to you guys. As we get closer, I'll say, hey, here's your choices. And then you guys can text me back or email me back and let me know what you prefer. And generally speaking, the majority will win. Asbury is here with WVSU gear on, so extra credit. So keep that in the back of your mind. Obviously, don't tell me now because I don't know what our time frame is, but once that happens, let me know when you want to do the exam. And it'll be just like it usually is. Online. Yeah, online starts at 8, ends at 8:50. And if you want to take a paper version, or if you want longer to take it, then you need to contact me sometime between now and then so we can make arrangements. The review, same thing. The question is when. When do you want to have the review? Just can't tell me that yet because we still don't know what our time frame is, but most likely the review will be sometime not during office hours. So that means you don't have to join me online for a review. You will just get extra credit points if you do. And again, you should definitely do the reviews. Hint, hint. Remember what happened last time with the reviews. So even if you don't come to this review, watch the video or watch my other videos. Anyway, speaking of reviews, remember the reviews are us covering the study guides, right? And the study guides are what I use to write the exams. So you're going to want to do the study guide sometime between now and when we do the review. That way, when we get together and we only have 50 minutes, you can hopefully write down the ones you need help with. All right, can you go to chapter 7, number 8, right? And I can give you the answers because I don't have time, or we don't have time in 50 minutes to cover every single question. So it would be good for us to narrow it down and distill it down to the ones you have problems with. So any questions about that announcement? Exams? All right, that segues great into independent work. So I'll remind you, please get some independent work done. Some of you, I'm so glad you finally started. It's a little late, but you still started, and you've seen that there's been some issues, like with the way you've written it and the way you've submitted it. And that's great, though, that you've done it now, and we've figured it out, right? So now you know exactly what you're supposed to do. And you know, like when you write something, it's not just like, hey, here's something, here's 10 points, right? It's 10 points if it's perfect and if it's the whole page. And that doesn't include quotation marks and things like that. So some people are going to wait until the last weekend to do this, not knowing how I grade, and then they're going to be like, oh, I didn't know, and then they're going to be out of luck, right? So I'm telling you, please, even if you have no intention of like working on it throughout the semester, and you know you're going to be one of those people that does it in the last minute, at least write one paper between now and then so you can see how I grade. Because again, mark my word, someone's going to screw it up, and they're going to get upset that they get a 0 for independent work because they're going to do it on the last weekend, and they're going to do it wrong. So yeah, write papers. You know, you can watch videos. And again, every time I send out an announcement, one of those, you know, attendance announcements, at the bottom of it, there's a little link that says, hey, here's a link. You can watch some videos for independent work. So any questions about independent work? That's just reminders. Again, I'll remind everybody, attendance is graded. In a sense, it's free points. So make sure you come. For everybody from now on, from here on out, everybody who physically comes gets a little extra credit for being physically present. So that's just giving you an incentive. Yes?
On my Google Classroom, chapter 6 and 7 doesn't have a study guide.
That's okay. If you look at the last announcement I posted, make sure you, that's another thing. Make sure you read my announcements closely. But anyway, the last announcements I posted, there's a chapter 5 study guide, and then there's a chapter 6 and 7 combined. A great question. And that you'll notice that that study guide is a little bit different than the rest. So I'm so glad you said that. So don't let that alarm you. And yeah, when you see that it's a combined 6 and 7 like that, don't, you know, now you'll know that's on purpose. It's not an accident. So anyway, yeah, extra attendance. Make sure you come. If you're missing anything, which you can see on the grade sheet, you know, you can go back and see which dates you're missing. Go back and watch the videos and do what the videos say. It's only half credit, but, you know, half credit is better than 0. So any questions about announcements and reminders? All right, before I start, let me look again on who's online here. All right, we got Hodges and Kip are the new ones. Kip and Hodges. Yep, all right. Some of the people who were online previously are now offline, but whatever. It's another good time to remind you. I said this yesterday or Monday too, but when things work properly, which usually they do, I get a spreadsheet from Google that tells me exactly who's online, what time they logged in, and what time they logged off. So don't share the attendance words and don't log in until you hear me say, oh, he's online, and then log off, because I'll know whether you were here or not. There's no need to cheat. It's only 2.4 points. If you can't make it, just watch the video, you know? It's okay. Anyway, let's get into it. If there are no other questions, let's get back into chapter 7, the last of the chapters for this exam. So to remind you, here's where we left off. We talked about something essentially useless. Your book included it, so did I. Well, the part that wasn't useless is we're talking about electrons getting excited, right? So electrons can get excited. That's a very unstable state, so they don't stay that way. They eventually fall back down to an unstable or to an unexcited state, and the process release energy. And one of the ways they release energy, sometimes they release light energy. And again, this is the useless part. There's no one, like, you know, I'm not going to ask you anything about fluorescent lights or, you know, glow sticks as important. What is important is what happens inside photosynthesis, inside the photosystems. And I've not told you what a photosystem is, so here you go. Actually, that'll be the first attendance word. Photosystems will be the first attendance word. If you're in person, you can send those words for extra credit. If you're online, send that word sometime before 9 a.m. for credit. If you're watching the video, assuming it records this time, and you're watching this later in the day, take a screenshot of me pointing at the word photosystems. Anyway, it's the photosystems where all this magic is happening, so to speak. But I am going to put it next to that, indicating there will be no questions about photosystems. Now, I'm going to use the word photosystems to describe how photosynthesis works. And I might use the word photosystems in some of the questions, not because the questions are going to be about photosystems, but because I'll be describing a situation. So again, you don't necessarily need to know what a photosystem is because there'll be no questions on it, but you need to know what it is just to understand what I'm saying and for the questions on the exam. Anyway, the thylakoid membranes, that's the part on the inside of the glass where photosynthesis happens. So it's almost like we're really zooming in, right? So first we said photosynthesis happens in leaves mostly. Then we kind of zoom in and say specifically it happens in all those organelles inside the leaves. Then we got even more specific and said it's actually in the thylakoid membrane, it's inside the chloroplast. Now we're getting even more specific and saying that there's these little things called photosystems that are within the thylakoid membranes, and this is where it actually happens. Each photosystem has a cluster of a few hundred pigment molecules. I'm definitely not going to ask you that either. On this next slide, I'm going to show you a visualization. So again, there's a big chloroplast, right? So a leaf cell has thousands of these, right? So we're looking at one of them, one of the thousands. Kind of cut it open. We're zooming into one of these thylakoids. Here's the thylakoid membrane, right? It's just like our membrane. This is a phospholipid bilayer. Right here, embedded in there, is the cluster. That's the photosystem, right? It's got a cluster of pigment molecules. And I use this analogy on Monday. It's a good analogy to help you remember this for the exam. But in a sense, you could think of photosynthesis as two parts. Well, it is. It's two stages. Powered. So that's the part one in solar powered, right? And the part, the ones we'll talk about later, that uses the energy collected from the power plant to actually make the glucose. So anyway, this is what we're talking about, right? We're still talking about the light reactions, specifically where it happens. So anyway, these things act as light gathering antennas, almost like solar panels. So what happens is the photon strikes a pigment, kind of like we talked about earlier, when the electrons get excited, right? So then not only are they excited, but then they start bouncing around in that excited state before they fall back down. And then finally they arrive at the reaction center. And the reaction center consists mostly of chlorophyll A and molecules that sit next to the primary electron acceptor, which traps a light excited electron. This is a lot of detail that you don't necessarily need to know. Basically what I said earlier is all you need to know, which is that photons hit an electron, they get excited, and then yes, just for your own background, just for your own knowledge, they do bounce around a little bit before they finally get to where they're going, which is the primary electron acceptor. So I'm going to put a big X to this, again, indicating that there's not going to be specific questions. Like I'm not going to ask you, what is the reaction center? What is a primary electron acceptor? I'm not going to ask you all that. There'll be no specific questions about those things. You just need to know those words because I'm going to use them in describing the process. Or if there's a test question that says, which of the two stages are electrons excited until they hit the electron or the primary electron acceptor, which I probably won't even say that. But anyway, that they might be part of the question, but they're not going to be directly questions about them. So anyway, at that point, another team of molecules uses that trapped energy to make ATP and NADH. I'll put another X to that. You do need to know that, but I'm putting an X to it because if you're taking notes right now, now's not the time to take those notes. This bullet point, we're going to spend a lot more time talking about. This is basically an overview. Everything that's just here, all this information I just gave to you really quickly, a lot of it. No worries. We're about to go back and talk about it in more detail. That's just an overview. We'll come back to it. So are there any questions so far? Okay. Here's a picture of what we're talking about. We already saw the left side of this picture, right? We're looking in the chloroplast. We're zooming into the thylakoid membrane. There's a photosystem. Now we're going to kind of like look at a bird's eye view. A photon hits one of those pigment molecules, probably chlorophyll B, right? Maybe one of those other ones we talked about. It doesn't matter for us or for the exam. It hits it. The electron gets excited. It's bouncing around. It just literally randomly bounces around until finally it gets to the last one, which is the chlorophyll A. And then that's where it gets basically captured, right? But you don't even need to know that much detail. As far as I'm concerned, you could just memorize this: the fact that light hits a pigment, it excites an electron, and while that thing's excited, it gets captured, right? Yes, it bounces around. Yes, technically, where it gets captured is the reaction center, and yes, technically that's chlorophyll A as opposed to the other ones. It doesn't matter. What you need to know is that in the light reactions, a photon hits the pigment, that excites an electron, and that electron is then captured in its excited state. And then obviously, as you could probably guess, that excited state, all that energy that it has is about to be put to use. So any questions about that picture? All right. So like I said, that excited electron is captured in its excited state. So let's learn about how we can use that energy to make ATP and to make NADPH. And again, to remind you, in a sense, as far as we're concerned, especially on the exam, that NADPH should be synonymous with excited electrons, right? So this bullet point could say how the light reactions generate ATP and excited electrons, right? We need ATP to build the glucose in the other reactions in the Calvin cycle. And we also need to incorporate high energy electrons. So those are the two things we need, right? Energy to squish the carbons together and high energy electrons to incorporate into the glucose. So here we go. There's actually two photosystems that do this. I'm going to teach them to you and they're in the order that they're in. But for the exam, you're not going to have to distinguish between one or the other. I'm not going to say, all right, which one happens between, which one happens in the first photo. Keep that in mind as I teach this. So when you're taking notes, you can leave that part out. So again, the photons hit the pigment, the electrons are excited, they bounce around, they finally get captured by the primary electron acceptor. And of course, electrons have to go somewhere, they have to come from somewhere. In this case, since we just lost an electron, it has to be replaced. So this is like I said on Monday, what gives it that new electron? It's the water. So that'll definitely be a test question. Where do the electrons come from in photosynthesis? They come from water. And in the process, that releases oxygen. Eventually, even though it's not on the screen yet, right? So water gave up that electron. It's going to get excited. It's going to go through some other stuff I haven't taught you about, but eventually it's going to get incorporated into the glucose. So again, that's the opposite of what respiration is. In respiration, we started with glucose, right? The electrons are in the glucose. We extract them, so to speak, from the glucose, and they release their energy until they are accepted by oxygen, which makes water, right? So this is just the opposite. Water says, here's the electron, plain old electron, you go ahead and get excited, and in the process, we produce oxygen. So yeah, test questions on this slide. Which of the two stages produces oxygen? Light reactions. Which of the two stages uses water? Light reactions. And then there'll be one question that has a three-part question, which is fill in the blank. It'll be something like, we release, blank releases oxygen and or blank releases electrons and produces blank, right? So water releases oxygen, or water gives up the electrons and produces. Any questions? So that excites the electrons, which are then just gone as far as we're concerned. Right now, for this discussion, they're just gone. They've disappeared. So we need to replace them. Where do they come from? The water. The water gives up some electrons. In the process, makes oxygen. Right? I'll put some X's through here, just like I did for respiration, to remind you. I don't even care about the numbers. Yeah, technically it's 2 electrons. Whoops. And technically it's that water, not only does it become oxygen, but it also becomes two molecules of, or two hydrogen protons, but I'm not worried about it. This becomes, releases oxygen. And those electrons are the things that get excited in the photosystems, and they're going to be put to use later. And that's what we're about to talk about. So any questions about this picture? All right, so we've got those excited electrons up there. And then that brings us to the second photosystem. So again, we have the excited electrons from the first photosystem. Where do they go, right? They can't just stay there. They have to go somewhere. And they're excited. And like I said earlier today, and the last thing on Monday, can't lose that energy. And they do. So in this case, the energized electrons, again, first photosystem, they go down an electron transport chain, just like in respiration, right? They go from. So essentially, here, I'd love to ask you, I'm still going to ask you, but the answer's right in front of you. I would ask, for this bullet point, when those electrons are going from photosystem 1 to photosystem 2, and they're releasing their energy going down the electron transport chain, what do you think we're going to produce? Of course, it's right in front of you, but the answer is ATP. And the reason I was hoping you would know that, even though I haven't taught it yet, is because it's the same thing that happens in respiration, right? We get those high energy electrons that came from the glucose in that case, and they go down the electron transport chain, releasing their energy to make ATP, right? Same thing here. We have an energized electron going down the electron transport chain, releasing its energy to make ATP. Here's a good question then, because it's not on the board. You should know it because it's the same as respiration. So how do you think these excited electrons going from protein to protein to protein in the electron transport chain, how is it making ATP? What is it doing to make ATP? See if anybody can guess. And again, the hint is it's the same. Electron transport chain that allows us to make a bunch of ATP. It's really important. The transport chain, and this thing just says, hey, it makes ATP. But it didn't show how at first, but then we got to it, right? So I guess I'll just let that marinate for a bit. I won't tell you yet. Hopefully somebody can come up with an answer. How is it that we make all this ATP from this process, right? But we'll come back to that. So anyway, again, from the beginning, light hits the pigment, that excites an electron, it gets captured in its excited state, and it gets then transferred to this electron transport chain, where it goes down, as in it's releasing its energy. That released energy is then used to make ATP. By the time it gets to this second photosystem, which again is just a bunch of pigments, right? It's lost its energy because that energy was given up to make ATP. So now it's not as energized anymore. So again, it gets energized again with more light, right? So then it's energized again. This time it gets accepted again. And now finally we have our high energy electrons that are then just taken away by that NADPH to be incorporated into the glucose. So again, this is what we're after. Stage 1 is all about this. ATP and high energy electrons. That's all we need. This is all what the light reaction is about, energy. We need energy in the form of ATP to squish all those carbon molecules together to make a glucose. And we need to incorporate some high energy electrons. So there we go. We have our ATP, we have our high energy electrons. And that complicated, but first photosystem, second photosystem, I don't care. It would be great if you can memorize the order in which this happened, but I don't care. The most important thing you understand is for the light reactions creating ATP, and you're harvesting high energy electrons. I mean, logically, it has to happen this way. Logically, if you were to first excite the electrons and then take them away, right, like this happened here, then you couldn't make the ATP, right? Because you would have already taken away those high energy electrons and incorporated it into the glucose, and you wouldn't have ever made ATP. So logically, it has to be this first, but again, for the exam, you don't need to know it in that detail. Or again, the fact that we're always dealing with 2 electrons at a time, right? I'm not worried about that much detail. Anyway, any questions about this picture before we talk about this? Okay, next picture. I like this one better. This is like a picture more like an analogy. So in this case, it's the same thing, but it's more like a cartoonish version. So here we have this non-energized ball, if you will, right? But then this person, boom, put some energy in there, right? And this is great because right now it has very low potential energy. In real life, if that was a ball, it has very low potential energy because it's right there next to the ground. This guy's giving it some kinetic energy, and then it goes up because of its kinetic energy. Now it's way up here, right? We got that primary electron acceptor. She's caught it. I assume it's a she. I guess I shouldn't assume that. This person with a ponytail has this thing. It's way up there now, right? So now it has a lot of potential energy, a lot more than it did when it was down there. So it gets put down on this thing and it rolls down the hill as it would, right? Because gravity is going to make it go from high energy to low energy, just like excited electrons go from high energy to low energy naturally. In the process, that energy is used to turn something, and turning something is making the ATP, right? But again, those balls are losing their energy because they're going downhill. Now they have less energy than they did here. Not that it matters for the exam, but they're just going to point out they're not all the way down where they started, but they're lower than they were here. So then again, we need some more energy to excite them. A photon hits them again, excites them again. Now they're really high energy, right? Even higher than they were here. And this is relevant, right? The locations are not on accident. So now we have these high energy electrons, and we have the NADPH, which is basically the electron carrier, the bucket, if you will, that's going to carry them off to the Calvin cycle so that they can be incorporated into the glucose molecule. So again, this picture is basically an analogy of the picture we just saw before this. I really like it. Yeah, and again, we still haven't even talked about this process. How is it that the electrons going down the electron transport chain make ATP? Does anybody want to guess yet? Going down the electron transport chain are pumping what against their hydrogen? I said it. They're pumping what against their concentration gradient? That's what we'll talk about. Hydrogen ions, just like photosynthesis or respiration. So in between the layers of those, well, in this case of the thylakoid membranes, right? Or in the case of respiration, in between the layers of the members, in between the layers of the mitochondria, we're pumping hydrogen ions in between them, right? They're going against their concentration gradient. So it's the opposite of diffusion. So it requires energy. From protein to protein, those proteins are using that energy to pump hydrogen ions against their concentration gradient. So now, trapped in between those two membranes, there's a lot of hydrogen ions, right? And what do they want to do? They want to diffuse down their concentration gradient because they've just been pumped against their concentration gradient. Of course, that part comes later. I guess I won't say that yet. But anyway, yes, let's just stick with that. Right now, on the slide, we're pumping hydrogen ions against their concentration gradient. And I like this. Your book points out the big difference between this and respiration. Because so far, it's the same thing, right? In respiration and in photosynthesis, we're using electrons to pump hydrogen ions against the concentration gradient. Main difference between this and respiration is that where are those high energy electrons coming from? For respiration, they came from food, or as we focus on, the glucose, right? In respiration, they came from glucose. Here, in photosynthesis, again, like we've already said, it's the water that's giving the electrons. So any questions about this side? All right, here we go. So again, this is almost like that respiration picture, but turned upside down. But the concepts are they're still the same, right? So we still have these electrons getting moving from protein to protein, right? So they're putting off energy. We're using that energy to pump hydrogen ions against their concentration gradient, right? So they're coming down here. So now there's a bunch of hydrogen ions in here. There's not that many out here. So what do they want to do? They want to diffuse back down their concentration gradient. So they do. And as they do, look, it's even the same enzyme, ATP synthase. It passes through. It literally turns something. You can't see it, but it's turning something like a turbine. And that's how we produce ATP. So again, for the exam, the most important thing here, even though that's a lot of information, for the exam, the most important thing you need to know about the light reactions is that we now have our high energy electrons and our ATP, both of which that are about to go to the Calvin cycle. That's how the light reactions provide high energy electrons and ATP to the Calvin cycle. Remember, the whole purpose of photosynthesis is to make glucose. And in that context, the Calvin cycle is where it's at. Everything's about the Calvin cycle, right? Because the Calvin cycle is the thing that's actually making the glucose. Light reactions, it's just providing the energy and the high energy electrons to do it. Let's see. The next word for attendance. How about this? I'll circle it and not say it. So that'll be the next one. I'll even draw an arrow to it. There's the next word for attendance. So again, if you're in person or watching online live right now, send those words before 9 a.m. And if you're watching the video, assuming it records, make sure you send a screenshot of me pointing at the word right there. It's circled and pointed to. Any questions? Okay. And again, like I keep saying, this is a lot like respiration. So I wanted to go ahead and throw this picture back up at you. This is respiration. It's not photosynthesis anymore. Look how similar it is, right? So how we have these high energy electrons that are going down an electron transport chain, right, from protein to protein to protein. And that in that process, they're releasing energy. That energy is used to push hydrogen ions against their concentration gradient. What do they want to do? They want to diffuse back down. Naturally, they diffuse back down the natural concentration gradient. That turns the ATP synthase. That makes a bunch of ATP. It's a good reminder, right? I go back. Photosynthesis, right? All right, so we'll talk about the Calvin cycle. Man, we might finish chapter 7 today. In which case, I mean, I'm definitely not gonna spring exam on you on Friday. So anyway, if we finish chapter 7 today, then the question is, when do you want to do the exam next week? In your mind, the later you take it, the more time you have to study. So just think about that, and I'll post the announcement later for you to make a decision. So anyway, to remind you, we just finished talking about light reactions. If you don't remember anything else about light reactions, remember, that's all about the energy, right? Calvin cycle is the thing that's what we're after, which is making the glucose. Light reactions, they're just providing the energy and the high energy electrons to the Calvin cycle to do their job. So here we go. Calvin cycle. Just like in a sense, the electron transport chain in respiration, that was the big one, right? That's making the most of what we need, right? We were after ATP in that process, and that's the one that made the most. So in this one, it's all about the Calvin cycle, so to speak. Technically, it regenerates a starting material with each turn. I'm going to put an X to that because you don't need to know that. That's just what makes it a cycle, right? That little bullet point there is just what makes it a cycle. Because you start with something, you add some stuff to it, you do some chemistry, you use it, and then again, you end up with what you started with. It's the cycle part. Obviously, each turn is going to have inputs and outputs, and we're going to obviously talk about those. And again, that is also something else that makes it a cycle. Not necessarily. Those two things together make it a cycle. Bullet point in of itself doesn't make it a cycle. What are the inputs? You need to know this. Carbon dioxide. Yes, from the air. I'm not going to ask you that. I mean, you know what? I don't even like that. And again, this comes from your book. Because as you know from your experiments, from those of you who have done the lab this week, sometimes the carbon dioxide is dissolved in water. It doesn't necessarily always come from air. But anyway, inputs, carbon dioxide, ATP, and high energy electrons. 3 test questions right there. Which of the 2 stages uses carbon dioxide? Calvin cycle. Which of the 2 stages uses ATP? Don't confuse that with makes it, right? Because Calvin cycle uses the ATP. Light reactions make the ATP. Which of the 2 stages uses high energy electrons or NADPH, right? Because that's the thing that's providing them. The one that uses them is the Calvin cycle. And again, it's the light reactions that produce it. So those are 3 test questions. And again, Calvin, cycle, carbon, all that goes together, even in respiration. citric acid cycle carbon, right? No matter what, whether we're talking about photosynthesis or respiration, you get a question that says which stage uses or produces carbon dioxide, right? If it has anything to do with carbon dioxide, the answer is going to be some sort of cycle, right? If it's respiration, it's Calvin cycle. I mean, if it's respiration, it's citric acid cycle. If it's photosynthesis, it's Calvin cycle. And here's another thing. I don't think I've told you this yet. All the questions for photosynthesis and respiration, for the most part, are going to be separate. So when I say which stage uses carbon dioxide, it's a multiple choice exam, all your choices are going to be photosynthesis choices, right? So it's going to be light reactions, Calvin cycle, both, or none of the above. So you won't have any test questions where I say which stage uses carbon dioxide, and then your options are Calvin cycle or citric acid cycle. Like, oh man, which one's which? You don't even need to do that. So again, if your question is about carbon, then the answer is cycle. Anyway, those are the inputs. What are the outputs? Well, this is what we're after. We're after glucose. So I'm going to put a circle through that because that's what you need to know. The outputs, as far as we're concerned, are glucose. That's how we teach it. But now I'm going to tell you that technically that's not exactly accurate. What it's really producing is this thing called glyceraldehyde 3-phosphate, abbreviated as G3P. That's a molecule that is then used to make glucose and other organic compounds. So you don't need to know that much detail. As far as we're concerned, again, photosynthesis creates glucose. You don't need to know about this little middleman, this G3P. But yes, like we said at the beginning of chapter 6 and also the beginning of chapter 7, and like you're going to learn later when we start talking about environmental things instead of cellular things, photosynthetic organisms produce their own organic compounds, right? That's why we need them in the ecosystems. And that's another good independent work topic that I've already mentioned. I'll mention it again. Like there's 20 amino acids that make up our proteins. So where do they come from, right? Some of them we can produce, some of them we can't. So that'd be a good topic. Which of the 20 amino acids can we make ourselves and which ones do we need to eat? Anyway, any questions about this? Calvin cycle uses carbon dioxide, uses ATP, uses high energy electron, produces glucose. All right. Here's a picture of what we're talking about. You don't need to know the name of this thing, but that is the acceptor molecule, because remember, it's a cycle, right? So that's the thing that's saying, all right, bring me the carbon, bring me the carbon. So that molecule that happens to have 1, 2, 3, 4, 5 carbons and 2 phosphate groups, not that you need to know that. It is then attached to the carbon from the air. And now here's the interesting thing. Then we end up with that 3-carbon molecule, which I have not named, and you can look up for independent work if you want. What is in the world is that thing called? Not that it won't help you understand any of it, but whatever. Side knowledge. So if you're a thinker, hopefully this picture is confusing you or pissing you off or bringing questions up. Like, what do you see anything wrong with this picture? Really look at what's happening here. We have a 5-carbon sugar that's being attached. We're adding another carbon to it, and we end up with that 3-carbon molecule. What's wrong with that picture? All right. How many math majors in here? None. Okay. Despite that, I bet some of you guys will get this question. 5, right? Because 5 carbons plus 1 is 1 carbon, carbon dioxide equals what? What's 5 plus 1? 6. Non-math majors. I'm not a math major either. So 5 plus 1 equals 6. 5 plus 1 does not equal 3. Right? So what? What happened? Also, where'd that other phosphate group go? Anyway, not that you're not going to know that much detail for the exam. I just wanted to point that out because, again, that's the kind of student I was. I'd be studying that. And at first, I would just memorize it like I'm supposed to. Then I'd start thinking about it. And then I'd be like, whoa, 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 that's not right. So I'll come back to that here in a second. Anyway, that 3-carbon molecule, right, we then use some ATP to do some more work to it. And we incorporate some high energy electrons. And then we still end up with this 3-carbon molecule that's called G3P sugar. And it's funny too, because you're not chemists, you're not biologists. Just like this thing. So you just have to take my word for it. They're not. the point here is, well, there we go. We made our G3P sugar. That thing has those high energy electrons incorporated into it. We finally used our energy from the light reactions at ATP. And here's where the whole 6-carbon thing comes back into play. So really what's happening is, right here, when we added this thing to this thing, right, like it shows right there, what we have is 2 of those 3 carbon molecules. They're just not shown in the picture, right? So whatever that thing's called, we actually have 2 of them. And then they both go through this whole process of the ATP and then NADPH. And now, at this part, we actually have 2 G3P sugars, right? So one of those G3P sugars, it exits, pieces out, right? And that thing is used to make glucose and other stuff. Of course, glucose is what we're interested in. And the other of the 2 goes back in. Some other chemistry happens to it. As you can see, you have 3 carbons. We do, add some more ATP. Then we're back where we started with this RUBP sugar. There's a lot left off. And again, I don't know, depending on the kind of student you are, that would irritate me. The fact that we went from 5 + 1 = 3. And here we go from 3 plus nothing, it gives us 5, right? None of that was accounted for. But unless you care about it, then it doesn't matter because I'm not going to ask you about it on the exam. That's a lot of detail to remind you what you need to know for the exam. And I'll put it in the form of test questions. Which of the 2 stages uses carbon dioxide? Calvin cycle. Which of the 2 stages uses ATP? Calvin cycle. Which of the 2 stages uses high energy electron? Calvin cycle. Which of the 2 stages produces glucose? Calvin cycle. And that's it. Don't need to worry about G3P sugar. You don't need to remember, like, I'm not going to ask you at what point is the ATP added and the the high energy electrons added. You do, and again, even though you do need to know that ATP is used, and by definition, if ATP is used, yes, technically, we're definitely going to produce ATP, but I'm not even going to ask you that. So again, we use carbon dioxide, ATP, and high energy electrons to produce glucose. There we go. That's the Calvin cycle. Any questions about that? All right, and this is another big picture. So now we're stepping back again. Now, even though we're pretty much done talking about the Calvin cycle, now we're looking at the whole photosynthesis process again. 2 stages of the photosynthesis, light reactions and Calvin cycle. And again, it's all about the Calvin cycle because it's the sugar that we're after, right? And then the light reactions, they're just there to support the Calvin cycle because the Calvin cycle needs energy to squish those molecules together. It also needs to incorporate some high energy electrons. And it gets that from the light reactions. So again, test questions. Which of the 2 stages uses light? Directly, right? And people are going to get this wrong, and it's going to make me sad because it's not a trick question. But which of the 2 stages uses light? Just the light reactions. It's in the name, not a trick question. Which of the 2 stages uses water? Light reactions. Which of the 2 stages produces oxygen? Light reactions. Which of the 2 stages produces ATP? Light reactions. Which of the 2 stages uses ATP? Calvin cycle. Which of the 2 stages produces high energy electrons? Light reactions. Which of the 2 stages uses high energy electrons? Calvin cycle. Which of the 2 stages uses carbon dioxide? Calvin cycle. And which of the 2 stages produces sugar? Calvin cycle. A lot of test questions on that one picture. Oh yeah, and also, when they are given up, what does that produce? Produces oxygen. Any questions about this big overall picture? If you could print that and write notes, that might be like the one flashcard you would need for photosynthesis. You could write everything you need on that one picture, right? Everything, every question I ask about photosynthesis is on that picture. Because I don't talk about photosystems and going from the electron transport chain or pumping hydrogen ions against a concentration gradient. I'm going to ask you all those questions about respiration. And I just assume if you know it for respiration, then you're going to know it for photosynthesis. So the whole pumping of the hydrogen ions and them coming back down the concentration gradient, I won't directly ask that as a question. I shouldn't anyway. I don't plan to for photosynthesis. So if there's no questions about that, let's say the last word for attendance is going to be sugar. And I'm going to throw a little curveball here, not for you guys. So you guys just send me the word sugar. If you're online, you're watching this video, instead of sending me the word sugar or me pointing at a screenshot of me pointing at the word sugar, find a music video on YouTube of a song that has the word sugar in it. There's a lot of them. And send that. Because it'd be pretty interesting if somehow two people had chose the same video. Anyway, any questions? That's great. We are done early. Keep an eye on your emails and your announcements. Start thinking about when next week you want to have the exam. Because yeah, we're done. We're done with all the material for exam 2. Also, yeah, not for you guys. For the recording, for the video, as always, if you're watching the video to get extra credit, you can like the video and take a screenshot that shows that you liked it. Because I feel bad that everybody who's in person gets the options to get extra credit and the people who watch the video don't. So that's the reasoning for that. One of the reasons, anyway. And again, if you're online, I know I can see some new names. So if you showed up late, just take the hit, get your fewer points. And then when I post the video, if you want, you can watch those to make up for the points you lost. But what you shouldn't do is try to get the words from people who were here the whole time. Please, that's cheating. I'll know if you were here or not. So please don't do that.